Today, I will be discussing understanding strengths and limitations of different benchmarking strategies using a variety of data types. My name is Sharon Liang. I'm a senior reviewer in the Division of Molecular Genetics and Pathology within the Office of In Vitro Diagnostics and Radiological Health at the CDRH FDA. I review molecular genetic tests. I'm also a member of the steering committee of the Precision Medicine Initiative project at FDA, overseeing bioinformatics working group. This talk is the fifth module of this course and will focus on benchmarking of NGS tests. As more and more NGS-based tests are used in clinical diagnosis, it is increasingly critical to ensure that tests are accurate and reliable. One way to measure the accuracy is to benchmarking during the test development. For NGS-based assays, benchmarking methods are likely to vary in their ability to evaluate different wet lab and informatics stages of a pipeline. It is helpful to understand how to create and evaluate benchmarking datasets and strengths and limitations of different benchmarking strategies using different data types. As you may well know, NGS-based tests are highly complex and involve a lot of steps. From DNA extraction to library preparation and sequencing to mapping and alignment and variant calling. With the involvement of databases, annotation, classification will be done for interpretation. Data analysis can span from base calling of the sequencer to interpretation. Along the test analytical workflow, benchmarking can happen in different steps. For example, reference materials can benchmark analyte types such as DNA. Datasets from either natural reference materials or synthetic datasets can be used to benchmark NGS bioinformatics pipelines. There may be other benchmarks in other steps. However, they are not currently available. This talk will focus on reference materials and natural synthetic datasets. As mentioned earlier, it's critical for sequencing methods and analysis pipelines to be technically accurate. Using this complex and rapidly evolving technology to detect genetic variants in even more complicated human genome is challenging. Variant calls can vary based on capture kit, sequencing platform, and bioinformatics pipeline. The performance of the test may depend on different uses, such as germline and mutation, somatic mutation detection are different. Specific to the data analysis part of the NGS tests, homologous sequences and structural variations challenge data analysis, and repetitive sequence challenge global mapping and local alignment. With current short-read sequencing technology, it's prone to false negative or false positive variant calls because of systematic sequencing errors, which you have learned in the third module of this course. So you know that benchmark data and evaluation methods are crucial. Then what are good benchmarks? First, we want different specimen types are all available in benchmarks. Then it's important to represent different genome context or regions. We like to see different variant types are included in the benchmark, such as small nucleotide, single nucleotide variants, small indels, fusions, and copy number variations. It would be great if there are difficult variants in different difficult regions. For developing clinical genetic tests, it's desired to benchmark medical relevant variants and also in a variety of ancestry groups. In order to create a well-known truth for benchmarking, multiple technologies should be used to characterize the sample in order to form high-confidence course. Last but not least, reference material samples must be homogeneous and stable. Up to date, the Genome in Your Bottle Consortium is the main body working on generating reference materials for NGS benchmarking. The initial work was focused on germline reference materials. One sample was extensively characterized, such as it was sequenced by different technologies, and a lot of groups have analyzed the sequencing data using different bioinformatics pipelines to generate high-confidence cores. However, it's not an easy job. The consortium generated a high-confidence core set that covers 70% of the genome with SNV and small indel cores, the easy-to-call variants. Then the question is how to get the rest of the 30% of the genome covered, and when the core set on other variant types, such as fusions, copy number variants, and large indels and deletions become available. The work is still ongoing, and the consortium has discussed the potential of generating new germline samples and new tumor normal pairing genomes for somatic mutation detection. The future reference materials will cover more desired characteristics that we have discussed. The reference materials that we just discussed are real data. Although it may be realistic, the features of the reference materials might not be perfectly characterized and may contain undefined sequence elements. Just as what we have discussed, whereas entirely synthetic data are clearly defined and characterized, they may not reflect all the features of natural human DNA sequences. Conversely, the community is using hybrid method that injects synthetic variations into natural sequences. This method is somewhat realistic and somewhat known, so each benchmark dataset has both strength and weakness. There are some tools available to generate benchmark dataset, such as Warsim, Hive in Silico, BAM Surgeon, and ART, and some comparator tools. They are all available on Precision FDA platform. The NGS community has put a lot of effort to create benchmark reference materials, datasets, and methodologies. Major players include Genome in Your Bottle, GA4GH, GetRM group from CDC, and Sequence Quality Control Consortium from NCTR FDA and Precision FDA team. The community, including clinical labs, is defining performance metrics, generating tools for easy comparisons of the final variant cause. People are also piling the list of critical clinical relevant variants and potential cell lines for new reference materials. In summary, it's important to develop benchmarks for designing and evaluating NGS-based tests and improving bioinformatics tools. However, there are limitations of benchmark datasets. Although there are different types of benchmarks, we need additional studies to compare natural data with different strategies for creating synthetic test sequences to understand the utility of various synthetic strategies. In order to benefit the NGS community in developing NGS-based tests, it's critical to continue the benchmark development. Thank you for listening to the introduction lecture. You are welcome to ask any questions to the emails listed on the slide. Thank you.